Imagine a small, unassuming fly. It's easy to overlook, just another tiny insect in the vast tapestry of life. It buzzes around, seemingly insignificant, a mere speck in the grand scheme of nature. But in the hands of Thomas Hunt Morgan, this tiny creature unlocked some of the biggest secrets of life. Morgan, a visionary in the field of genetics, saw potential where others saw only a nuisance. Morgan, a pioneer in genetics, dared to see the universe within this fly. He meticulously studied its patterns, behaviors, and genetic makeup. He used the humble fruit fly, a creature with a short life cycle and simple genetic structure, to unravel the mysteries of heredity. Through countless experiments and observations, he began to piece together the puzzle of genetic inheritance. His work revolutionized our understanding of how traits are passed down through generations. The fruit fly became a cornerstone of genetic research, a model organism that would pave the way for future discoveries. Morgan's journey began in the late 19th century, a time when the field of genetics was in its infancy. Scientists were just beginning to scratch the surface of how traits were inherited. The scientific world was grappling with the implications of Gregor Mendel's groundbreaking work on inheritance. Mendel had laid the foundation, but much was still unknown. But Mendel's laws, though profound, were abstract. They needed to be tested, proven, and expanded upon. Morgan sought concrete evidence, a tangible system to explore the mechanisms of heredity. He needed a living model to bring Mendel's theories to life. This quest led him to the fruit fly, a seemingly unlikely hero in the story of scientific discovery. Through this tiny insect, Morgan and his team would uncover the fundamental principles of genetics, forever changing the landscape of biological science. Thomas Hunt Morgan was born in Lexington, Kentucky in 1866. Growing up in a post-Civil War era, Morgan was surrounded by a community deeply rooted in history and tradition. His family had a rich history in the South, with ties to both the American Revolution and the Civil War. This heritage instilled in him a sense of curiosity about the past and a desire to understand the world around him. From a young age, Morgan displayed an insatiable curiosity about the natural world. He was fascinated by the intricate details of plants, animals, and the environment, often asking questions that went beyond his years. He spent countless hours exploring the fields and forests near his home, collecting specimens and observing the intricate details of nature. His explorations were not just a pastime, but a foundational experience that would shape his future scientific endeavors. Morgan pursued his passion for science at Johns Hopkins University. There he was exposed to the latest scientific theories and methodologies which further fueled his thirst for knowledge. He earned his PhD in 1890, focusing his studies on marine biology. His doctoral research was groundbreaking, shedding new light on the complexities of marine life and ecosystems. His early research took him to the shores of the Chesapeake Bay and the vibrant marine laboratories of the time. These experiences were crucial in honing his skills as a meticulous and innovative scientist. While his initial work centered on marine organisms, his interest soon shifted to the emerging field of experimental embryology. This new focus allowed him to explore the fundamental processes of life and development. This shift would prove pivotal, setting the stage for his groundbreaking work in genetics. Morgan's journey from a curious child in Kentucky to a pioneering scientist was marked by a relentless pursuit of knowledge and a deep-seated passion for discovery. After completing his PhD, Morgan accepted a position at Bryn Mawr College, a prestigious women's college near Philadelphia. This institution, known for its rigorous academic environment, provided Morgan with the perfect setting to further his scientific inquiries. At Bryn Mawr, Morgan continued his research in experimental embryology, focusing on the development of various organisms. His work involved meticulous observation and experimentation, often requiring long hours in the lab. He was particularly interested in the processes of regeneration and development, seeking to understand how organisms develop from a single fertilized egg into complex, multicellular beings. This line of inquiry was both challenging and fascinating, pushing the boundaries of contemporary biological knowledge. During his time at Bryn Mawr, Morgan began to delve into the work of Gregor Mendel, whose experiments with pea plants had laid the groundwork for the field of genetics. Mendel's experiments with pea plants, though conducted decades earlier, were just beginning to gain recognition in the scientific community. Morgan found Mendel's principles of inheritance particularly intriguing. Intrigued by Mendel's findings on inheritance, Morgan sought to explore these principles in his own research. He began to design experiments that would test Mendel's theories in new and innovative ways. This newfound interest in heredity would shape the course of his career, leading him to the doorstep of a scientific revolution. Morgan's work at Bryn Mawr was just the beginning of a journey that would eventually transform our understanding of genetics and heredity. In 1904, Morgan received an invitation that would change the trajectory of his career. Columbia University offered him a position in their zoology department. This move to Columbia provided Morgan with the resources and collaborative environment he needed to pursue his growing interest in genetics. At Columbia, Morgan established what would become known as the Fly Room. This small, somewhat chaotic laboratory housed on the top floor of Schermerhorn Hall became a hub of groundbreaking research. Here, Morgan and his students, including Alfred Sturtevant, Calvin Bridges and Hermann Müller, embarked on a remarkable journey to unravel the mysteries of heredity using the humble fruit fly as their guide. The fly room at Columbia University buzzed with activity. Morgan and his students meticulously bred generations of fruit flies, carefully observing and documenting the inheritance of specific traits. The fruit fly, with its short lifespan, rapid reproduction rate, and easily observable characteristics, proved to be the ideal model organism for their experiments. Through their meticulous observations, Morgan's team made a series of groundbreaking discoveries. They provided the first conclusive evidence that genes, the units of heredity, are located on chromosomes. They demonstrated the phenomenon of genetic linkage, where genes located close together on a chromosome tend to be inherited together. And they discovered chromosomal crossing over, a process where chromosomes exchange genetic material, further shuffling the genetic deck. In 1933, Thomas Hunt Morgan received the Nobel Prize in Physiology or Medicine. This prestigious award recognized his monumental contributions to our understanding of heredity. Morgan's work revolutionized the field of genetics, providing the foundation for modern genetics research. In a testament to his belief in collaboration and the shared pursuit of knowledge, Morgan shared the prize money with his former students, Alfred Sturtevant and Calvin Bridges. He recognized that their groundbreaking discoveries were the result of a collective effort, a testament to the power of teamwork and the vibrant intellectual atmosphere that flourished within the fly room. In 1928, after decades of groundbreaking work at Columbia University, Morgan moved west to California. He accepted a position at the California Institute of Technology, or Caltech, to establish a new division of biology. At Caltech, Morgan continued his research focusing on the role of genetics in development and evolution. Morgan's legacy extended far beyond his own research. He fostered a collaborative and rigorous approach to scientific inquiry that inspired generations of scientists. His students went on to make significant contributions to various fields, from genetics and developmental biology to evolutionary biology and medicine. The Fly Room at Columbia became a model for research labs around the world, a testament to the power of curiosity, collaboration, and the relentless pursuit of knowledge. The impact of Morgan's Fly Room resonates even today, nearly a century after its establishment. The principles of genetics uncovered by Morgan and his team laid the foundation for modern genetics research. Their work paved the way for advancements in fields such as medicine, agriculture, and biotechnology. From understanding and treating genetic diseases to developing new crops and therapies, the legacy of Morgan's Fly Room is woven into the fabric of modern science. His work reminds us that even the smallest of creatures can hold the keys to unlocking some of life's biggest mysteries, and it underscores the power of observation, experimentation, and the relentless pursuit of knowledge, driven by an insatiable curiosity to understand the world around us. Thomas Hunt Morgan's journey began with a simple yet profound question: how are traits passed down through generations? His search for answers led him to the humble fruit fly, a creature that at first glance seemed far removed from the complexities of heredity. But within this tiny organism, Morgan discovered a universe of genetic information waiting to be deciphered. Morgan's story reminds us that science is often a journey of unexpected discoveries. It highlights the importance of curiosity, observation, and the willingness to challenge existing paradigms. And it underscores the power of collaboration, mentorship, and the shared pursuit of knowledge. The legacy of Thomas Hunt Morgan's Fly Room, a testament to the enduring power of scientific inquiry, continues to inspire and shape the world around us.